welcome to this webinar, Higher Throughput Bioanalytical Methods Accelerate the Development and Evaluation of Advanced Biotherapeutics. I'm Lou Rahman, Publishing Director at DDW. Successful development of advanced biotherapeutics relies on extensive characterization and tight quality control. Characterizing binding properties of complex biomolecules is a critical step. Biophysical data combined with functional assays are needed to predict efficacy and potency, optimize drug delivery, assess critical quality attributes, and improve biologics drug design. Traditional approaches have limited use in a complex biological environment with multiple interactions, and to address these challenges, our speakers have developed and implemented better performing and higher throughput technologies. This event will be presented by Ron Morena, Manager in Analytical Bioassay at Tanvex CDMO, along with Dr. Neha Mishra, Senior Scientist in the Bioproduction R&D team, Revity. What you will learn: large molecule protein-protein interactions, measurement of binding activity using high throughput tools, assay development and implementation in the QC environment. Neha will introduce Revity's workflow solutions to support different stages of cell line development, starting with the CHO-source mammalian expression platform for expression of different molecules to bioanalytical tools that aid early-stage clone screening and protein characterization. Okay, I'll now hand over to Ron and Neha.
Today, I will be discussing the superior performance of alpha ELISA technology for measuring Fc receptor binding versus a traditional sandwich ELISA. Here's an outline of what I would like to cover today. First, I will introduce the company that I work for, and then I will discuss our lead antibody candidate's mechanisms of action, and discuss how Revity's technology assisted us in demonstrating biosimilarity by using their Delphia TRF kit for ADCC measurement and their Surefire Alpha ELISA kit for the detection of phospho-AKT. Then we'll be spending the majority of our time discussing Fc gamma R3A binding and the challenges we had using a traditional ELISA versus the benefits of switching to Revity's Alpha ELISA kit. I'll discuss results from our development, qualification, and our transfer to QC. And then I'll hopefully leave time for Q&A. From here on out, I'll be referring to Fc gamma R3A as CD16A. Tanvex Biopharma USA Inc. is a commercially and late-stage biosimilar company, and recently we added contract development and manufacturing capabilities. In-house we have manufacturing as well as process and analytical method development. But what are biosimilars? Biosimilars are highly similar to an already FDA-approved biological product and have no clinically meaningful differences in regards to safety and efficacy from the originator product. A biosimilar company must submit a similarity assessment for investigational new drug or biological license application. We have two biosimilar drugs in the late-stage FDA approval process. One approved in Canada and soon in the US. The other is a biosimilar to trastuzumab, which is specific to the breast cancer receptor HER2. The first step in assessing functional biosimilarity is to understand the drug's mechanisms of action. Let's just take a look at the basics. Here we see the Fab portion of our antibody binding to the HER2 receptor, consequently blocking the proliferation of cancer cells. Secondly, the Fc portion of our antibody is used to recruit our own immune system, i.e. natural killer cells, to kill the cancer cells via the Fc CD16A receptor. When testing for functional biosimilarity, these mechanisms of actions are the target of our test methods. Don't worry, I'm not going to go through them all with you today, but I will draw your attention to the methods in bold, and we'll be spending most of our time on the CD16A binding using the Alpha ELISA technology. Our lab was responsible for developing and qualifying over 15 methods, 3 of which we successfully used Revity's kits, and one of those 3 was validated for the batch release assay. Our first method of interest measures antibody-dependent cellular cytotoxicity, or ADCC, using Revity's Delphia time-resolved fluorescence kit. Let me walk you through this slide. First, we have our SKBR3 cells, which are HER2 positive. The cells absorb a fluorescence-enhancing molecule. ADCC is then induced by introducing NK cells and trastuzumab. The cells are lysed by ADCC. The cells are then pelleted by centrifugation, yielding a supernatant with fluorescent markers proportional to the number of cells lysed by ADCC. The europium is added to the solution to form a resolved fluorescence chelate, which can be measured with a properly equipped instrument. Signal strength is directly proportional to the amount of ADCC. Tanvex qualified this method, and we showed good accuracy, linearity, and precision. We were quite happy with that. Our second method measures the inhibition of the HER2 signal cascade using the Surefire alpha ELISA kit. When trastuzumab binds HER2, it triggers a signal cascade resulting in increased tumor growth. That signal cascade is shown on the right, and a key step in this signal cascade is the phosphorylation of AKT. By measuring phospho-AKT in lysates from trastuzumab-treated cells, we can measure trastuzumab's ability to inhibit the HER2 signal cascade. And how do we do that? We use Revvity's Surefire alpha ELISA kit for the detection of phospho-AKT. It uses an anti-coded donor bead and Revvity's Surefire anti-phosphoprotein-coded acceptor beads. The homogeneous format makes the assay easy to use and quantify. Before this kit, we had developed a live cell fluorescent imaging assay that required cumbersome staining of live cells, and it would have been very difficult to qualify. By comparison, this alpha ELISA was qualified and the assay data submitted to the FDA for similarity testing. Before we move on, I would like to explain the alpha ELISA technology a little further. We just saw it in the last slide, and this will be the focus of the rest of our discussion today. Alpha beads come in two forms, donor and acceptor beads. When the two beads come in close proximity to one another, the donor beads can be excited, causing a reaction that allows the acceptor beads to emit at 615. This is different than a typical sandwich ELISA, where you have to coat the plate and then wash the plate and then start sandwiching your reagents on top of each other and still having to wash in between each step. Using this technology, the beads are floating freely and come pre-coated already, and there are a multitude of options and configurations. This is great because you can add all the reagents in a homogeneous mix with no wash steps. Let's do a quick refresher before we move on. The two most important parts of the antibody are the Fab and the FC regions. The Fab regions contain the variable regions, and they give the antibody its specificity. However, in contrast, the FC region is largely conserved across all antibodies of the same class. The FC region is responsible for recruiting our own immune system, notably natural killer cells. The natural killer cells use the CD16A receptor to bind to the FC of the antibody causing ADCC. It is the binding of CD16A to the FC region of the antibody that is the focus of our third method of discussion today. Development of a 96-well plate-based sandwich ELISA was very difficult. Dose response curves were inconsistent, there was well-to-well variability, and it was very difficult to get a 4PL model fit. While we do get somewhat of an S-shaped curve, a sigmoidal-shaped curve, the R-squared values were lower than desired. And EC50 values, a critical parameter in determining binding potency, were variable run to run. Looking at the individual points, we see a big jump between the 0.1 and 1 nanogram per mil concentrations, and we see it again at the end of our curve. We were seeing this routinely regardless of the concentration series. This made 4PL modeling problematic. We also saw a lot of variability between replicates. In this bottom graph, I performed each concentration in multiple replicates to see the variability, and you can see that the replicates are now on top of each other. Also, there's variability in the maximum signal. In this bottom centigram, we have an OD of 1, and in the top centigram, we have an OD of 0.4. So at the end of the day, the precision and accuracy were poor, which made the method qualification very difficult. So what could be the root cause of that variability? May it be the transient and low affinity nature of CD16A's binding to the antibody's FC region? Let's dive into this a little bit. I performed these beta core binding and kinetic experiments in our lab here at Tanvex using our drug material. On the top, we see the FC region binding to CD16A, and in the bottom we have the Fab region binding to HER2. In the top sensogram, you can see that the FC binding occurs really fast. The association or the on rate happens really fast. The level's off, and the dissociation rate or the off rate happens really fast as well. In contrast, in the bottom sensorgram, the Fab portion of the binding, we have the on rate happening fast, but we have the off rate or dissociation happening really slow. Most sandwich ELISAs rely on that strong and specific Fab to target binding to form their sandwich and maintain binding throughout the integrity and the length of the assay and the wash steps. However, this does not appear to be holding up. Therefore, our scientific hypothesis was that possibly during all the washes in a typical sandwich ELISA, that things were washing off and we weren't getting that consistency that we needed to see to get good curve fits. Therefore, a homogeneous mix and read type of an assay should be superior and give us better sensitivity. Let's compare the sandwich ELISA workflow to the alpha ELISA kit workflow. On the left, we have the sandwich ELISA workflow, which has many steps, 7 to be exact, but more importantly, it has 5 wash steps, which we think is where the problem was. In contrast, on the right side, we have the alpha ELISA kit method, and all you have is 4 steps. All you need to do is do your serial dilution, add those samples to the opti plate, biotinylate CD16A, add that to the opti plate, add your donor and acceptor B mixture to the opti plate, and incubate for 90 minutes. And then read the plate with a plate reader that has the alpha module. Big difference in the two methods. On the right was our first experiment using the alpha ELISA kit. On the right, this 4PL curve fit was generated from our very first try with the kit. And you can see that I did the assay in duplicate, and the replicates are very tight. We get a great sigmoidal curve. We get an R-squared value of 1, which is the highest it can be. Comparing to our traditional sandwich ELISA, which I showed earlier, it's like night and day. We were very happy with this, especially when it was our first time out. This is a list of what is provided for you when you purchase the Revity CD16A Alpha ELISA kit. Having all the reagents provided for you is great. It saves us from having to go and check and qualify multiple vendors, which is what we would have had to do if we could even get the sandwich ELISA to work. On top of that, knowing the incubation time, the lighting conditions, the concentration of the reagents, and so forth, puts us that much further ahead in the development process. For example, just started baseball season. So if you like baseball, this is like getting a double, and all you need is a single or two to score a run, or in our case, to score a qualification. Even though this propels us further along than starting from scratch, there were some things we wanted and needed to check, because we knew eventually that this assay would be transferred to the QC lab and scrutinized by the FDA. Because so much was provided for us in using a kit, we were able to skip most optimization steps and focus our development on the specific needs of our QC lab. At Tanvex, we chose to use a 96-well half-area plate, an opti plate by Revity. We chose to go horizontally across the plate as opposed to vertically down the plate. Yes, we lose some throughput, but we felt having more points on the curve would be a better curve fit and give us a better sigmoidal shape. Also, we chose to do each concentration in duplicate. We also chose to put a reference standard and a control on each plate. And we also chose to use volumes that would be easily pipetable manually to have more accuracy. We also took this opportunity to look at our analysis software and move from Softmax Pro to PLA3, which is more preferable by our QC lab. We also chose to look at different lot numbers for robustness, specificity, and look at sensitivity to the differences in glycosylation or antibody. Our first attempt at dilutional accuracy testing during development went without a hitch, with the relative potency results very close to the target values. Take a look at the dose response curve on the left. As S curves are shifted to the left, it signifies less antibody is needed to achieve the same results, equating to increased potency. In green, we have our 200% sample, which is furthest to the left, and in blue, we have our 50% sample. In the middle, we have our reference standard, and a control at 100%, which are on top of each other as they should be. On the right side, I purchased a Fab-only antibody and ran that in the assay, and that is in orange, in purple here, and in blue we have a reference standard, which you can see there is no binding. Before talking about sensitivity, we should talk about glycosylation. For those unfamiliar, Fc glycans are carbohydrate-based molecules which help regulate humoral immune activity. The same cells manufacturing the same protein may glycosylate proteins differently based on the slight differences in the environment. We see here a glycan called G0. G0 is interacting with the CD16A to increase the affinity to the Fc region of the antibody. G0 is a glycan without fucose. It is known in literature that the higher the afucosyl glycan species, i.e. %G0, without fucose, can cause higher affinity for CD16A. Therefore, our question was, would we be able to see %G0 differences using the alpha ELISA kit? At Tanvex, we have a great analytical team that has developed methods to determine the %G0 glycosylation for different trastuzumab lots. See this table here? These samples have steadily increased percentages of G0 content, and as G0 goes up, so does the relative binding. Compare the 4PL fit of A to E, and you can see that the increasing %G0 brings the sample curve further left, increasing binding. Also, you can see how small looking shifts in the 4PL curve fit equate to almost doubling of potency compared to the reference standard, from 75% to 145%. This demonstrates why having an assay that reliably fits your model is crucial to produce the sensitive results needed to support similarity. In the far right graph, I took our drug product and I aglycosylated it, or I made an agly sample out of it, meaning I stripped it of its glycans and ran that in the assay, and this produced no binding. That is in orange, and the reference standard is in blue. Therefore, the answer to our question, can an alpha ELISA assay pick up %G0 differences? Absolutely. We easily moved through development to a protocol-driven qualification where we tested precision, accuracy, linearity, range, specificity, and sensitivity with successful results. That's a lot of S's right there. This was key for us because we did not intend to use this assay in an R&D setting. We aren't screening for certain antibodies, and we are not using high throughput screening. These attributes and qualification are very important to use because we wanted to present our similarity data to the FDA, and we knew the assay is going to be passed on to QC for a release assay. Intermediate precision and repeatability are very well below the 10% GCV. Accuracy results centered close to 100%, and then we qualify the assay over their entire tested range of 50% to 200% compared to the reference standard. And as in development, the alpha ELISA successfully demonstrated specificity and sensitivity. Lots of S's again. Looking at accuracy and linearity plot, you can see that the samples tested at the different accuracy levels center around their respective targets. Here we have the target, here we have the measured, and here we have the percent recovery. Percent recoveries deviated less than 10% from the target. As far as accuracy and linearity testing goes, these are pretty good results and well within the goals set forth by our qualification protocol. In summary, we successfully replaced our CD16A sandwich ELISA that had many wash steps and took much longer to do with Revi's alpha ELISA kit. The assay length went from 2 days to a half a day. You can easily perform multiple plates in the morning, and in the evening it is that quick and easy. The data now fits a standard 4PL curve fit really well, improving the accuracy and the precision of our results. For us, a big positive was that we were able to submit this data to the FDA for similarity testing, and they had no questions asked. In the end, Tanvex used this kit method to speed through development, qualification, and quality control method transfer and validation as easy as driving on autopilot. We submitted alpha ELISA data to the FDA to show similarity as part of our BLA, and currently use the alpha ELISA as part of our QC batch release testing. Before we move into Q&A, I would like to take a moment for acknowledgments. I would like to acknowledge Patrick Esperon, who has been exceptional at communicating with us. He would ask in advance how many kits we would need and when we would need them. They would talk with his manufacturing team to find out when they would be available. So it was a good back and forth, open communicative relationship, and it still is. Patrick is great at fostering a collaborative spirit. Also, I'd like to thank the whole Revity support team. They are always very responsive to our needs and very knowledgeable in what they do. I would like to thank my supervisor, Dr. Anka Hartung, for all her help. She not only was an analyst on this project, but she was a statistician as well, crunching the qualification data, setting up a template in PLA3 to analyze the data, training me in PLA3, and helping me set up all the assay and sample criteria, and basically guiding me along the way and transferring this to QC. I'd also like to thank Michelle Neelan for her help with the pre-qual, qualification, and similarity testing. In the similarity testing, we tested over 35 samples in duplicate, quite a lot. Then I would like to thank Dr. Hongwei Yun for working with me in getting this transferred to the QC team and for his validation work on this assay. And I would like to give a special shout out to my colleague, Dagan Acevedo, for further similarity testing work and help in completing this slide presentation. Lastly, I would just like to mention that we Tanvex are a full-service CDMO. As you can see, we have a great working relationship with Revity. They have an alpha ELISA kit for all the Fc receptors and many others, as well as 200 plus targets using the Surefire kits. Or if there's an interaction that doesn't seem to be working for you, but you think an alpha ELISA would work, then they can work with you and make you a custom kit. Then if you don't have the personnel or instruments to perform the work, Tanvex would be happy to do that work for you. We are trained and have operators ready to go, and we have 3 Envisions. Thank you for your time. At this point, I'd be happy to answer any questions you might have. Thank you.
Hello everyone. I'm Neha, a senior scientist from the bioproduction team at Revity Cambridge. Today I'm going to present some work where we have specifically utilized the LabChip instrument for product quality assessment. Today, I'll first introduce Revity's support solutions for cell line development, including the ChoSource expression platform. I will then present 2 different case studies where we have applied the LabChip GX2 instrument for product quality assessment of proteins using the ChoSource platform. The first case study involved N-glycan analysis of an IgG1 molecule expressed using ChoSource cell line, and the second study will explain how we expressed a complex molecule using a ChoSource cell line and analyze the product quality of this on the LabChip. First, I will begin with an introduction to the ChoSource expression platform. Revity Bioproduction supports cell line development with the ChoSource expression platform that includes cell lines and expression vector sitting upstream of this pipeline shown here. Revity also provides solutions downstream of this by offering tools such as the Celaca for cell counting, Celigo for monoclonality verification, and the LabChip for product quality assessment, amongst others. The ChoSource expression platform sits at the core of Revity's bioproduction offering. It includes our GS knockout cell line built on the ChoK1 strain with proven performance in expressing many different types of biopharmaceuticals. Our ChoSource GS knockout offers full flexibility to licensees and can be used with any media or feeds. It has a comprehensive cell history documentation and protocols supporting cGMP clinical and commercial applications. Our platform has been extensively used worldwide with over 100 licensees to date and more than 60 INDs and regulatory filings in different territories. We also have another host cell line based on our ChoGS knockout, the ChoSource ADCC Plus cell line. This cell line is capable of producing antibodies and Fc fusions lacking fucose, which promotes enhanced antibody-dependent cell cytotoxicity effector function, and can be used to generate more potent therapeutics in areas such as oncology, anti-infectious, or autoimmune conditions. Finally, we have the ChoSource TNT transposon technology, which is a 2-component transposon-based platform with very robust performance compared to the standard random integration technologies. As I mentioned earlier, I'll be presenting a case study where we have utilized the lab chip for N-glycan analysis of an IgG1 molecule that has been expressed using the ChoSource expression platform. Before I explain the N-glycan analysis, I would like to first discuss how we use the ChoSource TNT transposon technology for expression of an antibody. TNT transposon technology includes a transposon vector and the TNT transposase mRNA. The vector contains a dual expression cassette for expression of up to 2 genes of interest. It also includes an optimized glutamine synthetase selection marker. Both the TNT vector and mRNA are co-transfected together into the ChoSource host cell line, where the transposase is expressed and binds to the terminal inverted repeats. The enzyme then excises the transgene out of the vector and integrates the transgene into the genomic region. A typical cell line development workflow for expressing an IgG antibody would look like this. The first step is molecular cloning, where the vectors containing the gene of interest are designed and then the cloning is performed. The vectors are then stably transfected into a ChoSource cell line, in this case the GS knockout cell line, using electroporation. The transfected pool of cells then undergoes selection by adding glutamine-free media. We do not use any other form of selection during the bulk pool selection process. Once the cells recover from selection, we then perform a copy number analysis using ddPCR to detect the number of copies integrated into the genome and also perform a 14-day fed batch overgrowth experiment to assess the titer. Lastly, we perform product quality analysis such as N-glycan analysis, charge variant, and aggregation analysis depending on requirement. We use Revity's LabChip GX2 high-throughput instrument for product quality analysis. LabChip is a microfluidic capillary electrophoresis-based instrument for protein characterization, which can be used instead of STS-PAGE and conventional CESTS. Assays on LabChip can be performed under reducing and non-reducing conditions, and the results can be conveniently analyzed by viewing on electropherogram, tabular, or virtual gel formats. You may ask, why do we need to analyze the N-glycan profile for our IgG1? N-glycosylation is the most common post-translational modification that is found on IgG1s and is considered a critical quality attribute. These modifications are important as they can affect efficacy, stability, PK/PD, and so on. There are many factors that may affect N-linked glycosylation of an antibody, such as the cell culture processes used, the media used, methods of manufacturing, as well as the cell line used for the expression of the IgG1 antibody. Therefore, it is important to assess the N-glycan profile of antibodies produced using the ChoSource platform. So we performed N-glycan analysis for our IgG1 that was expressed using ChoSource TNT transposon technology. We collected supernatant from one pool and 5 different clones to compare. Samples were taken from day 14 of the fed batch overgrowth experiment, which was then protein A purified, and then post-sample prep, analyzed on the lab chip. As you can see from the profile here, all major glycans are identified as expected, and the N-glycan profile between pool and clones are also similar. This shows that the IgG1 expressed using ChoSource TNT technology displays the expected N-glycan profile, and therefore we can accelerate our pipeline by using pool material for early studies. With the help of LabChip, we also showed that the ChoSource platform is robust and can be used to produce antibodies with good quality. In the second case study I'm going to present today, we expressed a 4-chain bispecific antibody using ChoSource TNT transposon technology and performed quality analysis of the antibody using LabChip to determine and select the pool with the right conformation of the bispecific. Before I get into the details of the study, I would first like to introduce bispecific antibodies. Monospecific antibodies such as the one shown here have 2 identical antigen binding sites and bind to one specific antigen. In contrast, Bispecific antibodies are non-natural engineered antibodies that have two different antigen binding sites. This allows them to bind to two different antigens at the same time. These antibodies can be used to bring two different cell types together or target two different antigens. This means that the cell now requires 3 to 4 different chains to express one single antibody, leading to multiple incorrect forms that can form within the cells if the chains randomly combine. Newer technologies have been used to engineer these antibodies. A few of these are knobs into holes, charge pair, or seed body. Knobs into holes introduces a bulky amino acid on one of the heavy chain and a smaller amino acid on the other, avoiding pairing of the same types of heavy chain. In the work that is being described here, CHO-source TNT transposon technology was used to express an asymmetric 4-chain bispecific antibody in the CHO-source GS knockout host cell line. The antibody consists of two light chains, light chain 1 and light chain 2, and two heavy chains, heavy chain 1 and heavy chain 2. The design of the antibody avoids most of the incorrect forms, but the two common contaminants in this type of antibody expression are the knob-knob and the hole-hole homodimers. Since the homodimers have a molecular weight very similar to the heterodimer molecule, it is almost impossible to remove these contaminants. I have shown earlier in the presentation that an antibody can be expressed using the CHO-source TNT by using a transposon vector and the transposase mRNA. However, for a bispecific antibody such as the one I just showed, the chains are cloned into two different vectors and then co-transfected into the host cell line, and GS selection is used for selecting transfected cells. Here I'm showing the bispecific antibody expression workflow in a bit more detail. First, 8 different vectors were designed to contain the 4 different chains of the antibody so that all positions are tested. Next, the vectors are stably co-transfected by electroporation into the GS knockout cells. Post-transfection, the cells undergo selection by using glutamine-free media, and then we perform copy number analysis to assess number of integrations of each chain, and we also perform a fed batch experiment to assess pool productivity. A product quality assessment is then performed using LabChip. These are the vector configurations that were designed and expressed in GS knockout cells during this particular study. Over the next few slides, I'll present some data that we have obtained over the duration of this study. To generate pools expressing all 4 chains of the bispecific antibody, the 8 vectors were co-transfected in pairs in a 1:1 ratio, vector 1 is to vector 2, into the CHO-source GS knockout host cell line, along with the CHO-source TNT transposase mRNA. 2 pools were generated for each co-transfection. The transfected pools were subjected to selection in a growth medium without L-glutamine. The co-transfected pools recovered from selection over 8 to 16 days. The different recovery times suggest that the antibody chain position within each vector has an impact on the selection recovery of the pools, possibly due to the varying expression levels of the antibody chains in different positions. Copy number analysis for each pool was performed to detect the number of integrated copies of the individual antibody chains and GS gene. When we looked at integration of each chain present in one vector, equal number of integration was observed, which confirmed that the transposase integrates the intact transposon. For instance, heavy chain and light chain 1 in configuration 2 are integrating in equal numbers. Since the vectors were co-transfected in a 1:1 ratio, an equal number of copies of the 4 chains were expected to be integrated in each pool. Vector configurations 4, 7, and 8 had an almost equal number of all 4 chains. However, in other conditions, the integration of the 2 vectors was not in a 1:1 ratio. For example, in configuration 1, the vector containing the knob heavy chain integrated fewer copies than the vector with the whole heavy chain. This might be since increased production of knob-knob homodimers could aggregate within the cell and cause cell death. Consequently, cells integrating more copies of knob heavy chain containing vectors may not survive post-transfection. The observed copy number of the GS gene was higher than that of the individual heavy and light chains, as it is integrated from both vectors. For all pools, the GS gene copy number closely matched the sum of the average copy number of the chains integrated on each. This further demonstrates that the CHO-source TNT transposase mRNA integrates an intact transposon consisting of both expression cassettes and the GS gene. The antibody expressed by all pools was further purified and analyzed in non-reduced conditions on the LabChip GX2 Touch system and compared against the reference bispecific antibody. The analysis performed with denatured samples allows proteins to be separated on the basis of their size. This enables the identification of different conformations of the antibody, such as heterodimers, homodimers, glycosylated variants, and free chains. When the chromatogram peaks were compared for all configurations, configuration 8 showed the denatured profile that was the most comparable with the standard. Next, I'm showing the analysis performed with reduced samples. This allows for the identification as well as quantification of the individual antibody chains. Although conditions 4 and 7 showed integration of all chains in a 1:1:1:1 ratio, they did not display the same ratio of chain expression. For example, condition 4 showed lower expression of heavy chain 2 and light chain 2 chains as compared to heavy chain 1 and light chain 1 chains. However, antibody produced by configuration 8 pool exhibited 94% correct conformation as compared to the reference molecule with light chain and heavy chain levels, which was similar to the reference standard. Therefore, by incorporating quality analysis on the lab chip early on in the cell line development process, we were able to select the pool with the correct conformation. In conclusion, today I have shown that we were able to integrate the LabChip GX2 in our CLD pipeline for product quality assessment. We were able to perform N-glycan profiling for an IgG1 antibody and showed that the antibody displays the expected glycan profile for a mAb. By analyzing both pool and clones derived from this pool, we also show that the glycan profile between pool and clones is similar, and therefore pool material can be used for early studies. Further, we were able to reaffirm the robustness of our CHO-source expression platform. We also analyzed a bispecific antibody molecule that was expressed using the CHO-source expression platform, and we show that we were able to produce this antibody in its correct conformation as compared to a reference molecule. In this study, we used LabChip to analyze the product quality of the antibody produced and were able to select the best pool with the correct conformation. Today, I have shown you that Revvity's CLD workflow solutions can enable and accelerate critical phases of cell line development. Thank you for listening, and I'll be happy to take any questions now.
Thank you, Ron and Iha. Those were excellent presentations. Before we go over to questions, I'd like to remind everyone that this webinar will be available to watch on demand at www.ddw-online.com. Okay, we'll go over to the questions then. And the first one that we have is, why does the curve change shape from the traditional ELISA to the alpha ELISA?
The alpha ELISA is a competition-based ELISA. Therefore, the sigmoidal curve direction is switched, meaning the highest concentration where the most competition is happening is at the far left of the x-axis, which is the inverse of a traditional sandwich ELISA.
Great, thank you for that answer. Second question, did you see lot-to-lot variability between the alpha ELISA kits, and did you test multiple lots for the CD16A?
We tested multiple lots of Revvity's alpha ELISA kits and various sizes as well. I did not see any lot-to-lot variability within our final results.
Thank you, that was a great answer. And the next question is, can Revvity CD16A alpha ELISA assay be used in a higher throughput manner?
Yes, in fact, the package insert is written with a 384 well plate example. Also, the Envision instrument can be fitted with a stacker to feed the plates into the instrument. I would suggest talking with Revvity because they have robots and the right instrumentation to use high throughput screening.
Thank you for those responses, they were excellent. Okay, Neha, we'll now go over to you for some questions. And the first one that we have is, what are the advantages of using two separate vectors for the expression of bispecifics?
Using two separate vectors can give us the flexibility to play with the ratios of each vector and also to test different chain positions to tweak the expression of each chain to identify the correct conformation. In the example I discussed today, the molecule needed equal number of integrations and expression of each chain, and this may not be the case for all molecules. Some molecules may require an excess of 1 to 2 chains, and having the possibility of having different ratios is a good way to achieve that goal.
Okay, thanks, Neha. That was a great answer. And the next question is, what percentage of correct conformation do you expect when expressing a bispecific antibody?
Usually at the pool stage, generally we would expect more than 90% of correct conformation. In the study that I presented today, we had 94% of correct conformation when compared to the reference molecule. And this was at the pool stage, which is what we expected. This high percentage at pool stage is a good indication that the clones derived would have a high percentage of correct conformation as well. And this is why it is important to include the quality analysis early on in cell line development.
Thanks, Neha. And then the last question is, what is the advantage of using lab chip for N-glycan analysis over other methods such as mass spectrometry?
First and foremost, the high throughput automated analysis is a major advantage over other methods. Usually, a lot of different samples are generated from cell line development campaigns, and they need to be analyzed in a timely manner as the data is important for deciding next steps in the campaign. By implementing LabChip for such large number of samples, it makes it less labor intensive than, for example, using mass spec for glycan analysis or STS page for aggregate analysis. Also, STS page is semi-quantitative, whereas LabChip provides quantitative protein analysis. Data can also be reviewed in a user-friendly format, such as via the virtual gel, electropherogram, and table summary formats. Another advantage is the low sample quantity requirement, which is also a great advantage as sometimes during screening stages we do not generate huge quantities of protein, and therefore by using LabChip it's quite helpful in this scenario. It can also be cost saving because of this reason.
Okay, thank you, Neha, and thank you again, Ron. Those responses were excellent, as were the presentations. And thanks again for talking to DDW today. I'd just like to remind everyone that you can listen to this webinar again on demand at www.ddw-online.com. You can now view Drug Discovery World content online or on your mobile or tablet. Thank you for listening.